So it is now my pleasure to let Wayne off the hook and introduce our next speaker, Dr. Daniel Liptzin, who is a research soil scientist with the Soil Health Institute and has a background in biogeochemistry and nutrient cycling. Currently, Dan is working on dairy, soil, and water regeneration, where he provides technical leadership for measuring greenhouse gas emissions at multiple sites across the U.S. Previously has served as a project scientist for the Soil Health Institute in the North American project to evaluate soil health measurements that Wayne talked about in detail. And currently, his research and interests are focused on greenhouse gas emissions, soil carbon, nitrogen dynamics, and soil health methods. And Dr. Liptzin will be sharing some effective measurements for assessing management impacts on carbon and nitrogen, which is, it feels like one of the major topics of the day. So I'm very excited to hear this talk. So again, I'll be back. So please continue to put your questions in the Q&A section.
Well, thank you for that introduction, Lakeisha. And thank you all for being here today. My name is Dan Liptzin, and I'm a research soil scientist at the Soil Health Institute. And I'm going to be talking today about our work on soil health measurements related to the carbon and nitrogen cycle. And all of the data that I'm going to be talking about today is from the North American project to evaluate soil health measurements that Wayne just introduced. And so just as a reminder, this was a project where we collected soil samples from a depth of 0 to 15 centimeters using the same field methods, same sampling methods from more than 2,000 plots representing 688 treatments at 124 sites in the major agricultural regions of North America. And we did this to identify effective, widely applicable measurements of soil health. And this work was only possible thanks to our partnering scientists, many of whom are shown here on the bottom, who volunteered to let us sample their existing long-term experiments that contain treatments that could be used to test the effects of adopting soil health practices. And we're interested in carbon and nitrogen cycling because they're important soil functions. However, there's no one single measurement that we can take to quantify carbon cycling in soils. It's possible to make measurements of the amount of different types of carbon in the soil, so the soil organic carbon or the microbial biomass. It's possible to measure the rates of processes like the production of CO2 from organic carbon. But in general, we consider healthier soils to be the ones that have bigger carbon pools and higher rates of these processes. And I'm not going to talk about it today, but the results of NAPSHM suggest that any management that increases the amount of carbon that gets added to the soil, whether it's through amendments like manure or compost, whether it's through increasing photosynthesis, growing more biomass that leads to more litter addition to the soil or more root exudation, or just retaining that residue in the system, all of those practices that increase organic input lead to higher values of all of the carbon and nitrogen measurements that we took today, that we took in this study. And the relationship between nitrogen cycling and soil health is parallel in many ways. The key inputs are the plant residues, also the animal manure, so these organic inputs. Nitrogen has the difference that we're also tend to add commercial fertilizer sources to nitrogen. But once again, we can look at the pools of nitrogen. So we can look at the nitrogen in the soil organic matter. We can look at the inorganic pools of nitrogen, like ammonium. But often what we're interested in is the processes. So the change from organic matter to ammonium, a process we call nitrogen mineralization. And so as Wayne mentioned, there are more than 30 soil health measurements that were part of NAPSHM that were included in this study. And later in this session, Diana Bagnall is going to talk about these water and structure indicators, and Liz Reiki is going to talk about some of these microbial community indicators. But I'm going to focus on the carbon and nitrogen pools and processes. And so one big advantage of NAPCHM was that we actually did all of these measurements ourselves. We collected the soils, we handled them the same way, we shipped them to the same labs. And so the same exact, when we say soil organic carbon was measured on the soil, the exact same methodology was used on all of the soils. So in terms of carbon measurements, we looked at the 7 here shown on the left. We measured the soil organic carbon by dry combustion. So this is the total amount of organic carbon in the soil. We measured permanganate oxidizable carbon. This is a pool of carbon in the soil that's defined by its chemistry. So it's carbon that can be oxidized by a permanganate compound. We looked at 2 measurements of potentially mineralizable carbon. One of them is a 24-hour incubation, and one of them is a 96-hour incubation. And you might be familiar with them because the 24-hour one is part of the Haney test, and the 96-hour one is part of the Cornell assessment of soil health. We looked at the activity of the beta-glucosidase enzyme. This is an enzyme that results in the cleaving of glucose from larger carbon molecules, and glucose, of course, is a good energy source for microbes. We looked at the water extractable organic carbon, and this is a pool of carbon defined by its chemistry again. So it's the carbon that was either in the soil solution or so loosely sorbed to the soil that it can be removed just by water. And then finally, we looked at loss ionization, which is really a measure of soil organic matter. But it can be converted to carbon using people convert it to carbon by using a fixed ratio of carbon to organic matter. And for those who are interested in learning more about these carbon indicators, there is a journal article that came out earlier this year in Soil Biology and Biochemistry that goes into all the details of these measurements. And then I'm including this one that was in the microbial community analysis, but the microbial biomass measured by phospholipid fatty acid is also a measurement of a soil carbon pool. So we can measure the carbon in the microbial biomass in the soil. And there's a paper that's been submitted to Applied Soil Ecology that largely focuses on the individual biomarkers from the PLFA analysis, but we also do report on the microbial biomass in that paper. And one important difference when we're talking about PLFA analysis compared to all of the carbon and nitrogen indicators I'm going to be talking about is because it is a microbial community analysis, it was done on soils that were freeze-dried right away to preserve that microbial community. In terms of nitrogen indicators, there were 5 that we looked at. There's total soil nitrogen analyzed by dry combustion. So this is actually measured simultaneously with the soil organic carbon. We looked at the autoclavable citrate extractable proteins. This is another chemically defined pool. It represents a fraction of the total nitrogen pool that it's supposed to represent protein in the soil. We looked at the potentially mineralizable nitrogen. This is representing a process related to nitrogen in the soil, the amount of nitrogen that's being converted from organic forms to ammonium during a 7-day lab incubation under anaerobic conditions. We looked at the enzyme activity of N-acetyl beta-glucosaminidase, and this is an enzyme that breaks bonds in a large molecule, in this case chitin, to produce amino sugars, which are also compounds that provide a source of nitrogen and energy to microbes. And then finally, the water extractable organic nitrogen. So this is done with the same extraction as the water extractable organic carbon, but in this case, we measure the nitrogen on it. And there's a manuscript that should be submitted any day looking at these nitrogen indicators. And I'm going to skip ahead a little bit to why we didn't choose a few of these measurements before I get to showing most of the data. And so we get a lot of questions about loss on ignition because there's a long historical record of comparing loss on ignition to soil organic carbon. And our data set shows the same thing. These two measurements are highly correlated. The line here represents the best fit between them. But one of the issues with loss on ignition is that it's sensitive to the clay content in the soil. And so the colors of the points on here that represent the treatment means of all of these samples are the clay content. And what you'll notice is that above this best fit line, you see most of the blue and purple points. So those are the soils with high clay content. And below the line are the orange and red points that are the soils with low clay content. So by measuring loss on ignition, you can introduce a bias into your analysis where the clay content affects the results. So we prefer not adding that uncertainty into the interpretation to the measurements. And so I'm not going to show you the plots of loss on ignition, but as you might guess from the high correlation with soil organic carbon, it does tend to respond similarly to soil organic carbon. Another measurement that I'm not going to show on the plots is the microbial biomass from the PLFA analysis. Once again, the response is similar to the other measurements, but because the samples must be freeze-dried right away and very few commercial labs perform this measurement, it makes it more suitable for research purposes than for widespread use at scale. For those of you who have done these measurements at all, I just want to throw up a quick view of what these measurements look like. So these are histograms for the 6 indicators I'm going to talk more about for carbon. So just to, and then these values represent the mean values from the analysis, just to give you a quick picture of what the data looks like for carbon. And then a quick picture of what it looks like for nitrogen. Once again, with the mean values shown on these plots. So the first step in determining the effective, widely applicable measurements of carbon and nitrogen cycling is to understand how those values that I just showed you varied in the data set. So there was a wide range of values, and we want to understand why they vary. And so, as mentioned before, we collected these samples across a wide range of climates. So cooler and wetter sites in the blue and hotter and drier sites shown in the red colors here. But we also collected them across a wide range of soil types and a wide range of managements. And so we wanted to evaluate how these measurements varied across sites. So for example, here's an experiment in North Dakota. Here's one in Texcoco, Mexico. And so we wanted to know, do the absolute values of these measurements differ across sites? Because if they do, then site needs to be considered in order to interpret these measurements to put them in context. We also looked at the variability between sites. And so we can look at the differences between, the different treatments, the site in North Dakota. We can look at the differences among the treatments at the site in Mexico. And only measurements that respond to soil health practices are really useful in this case. So in these two examples, we'd be looking at how big the change was associated with tillage and crop rotation. And so we evaluated where the variability was using an analysis called variance partitioning. And so when we do that with the carbon measurements, we find that most of the variability is among sites, from 62 to 80% of the total variance in the data set is among site variance. So site is really important. But then when we're looking at management, what we're looking at is this 9 to 29% of the variance that's associated with differences among treatments within sites. And you're going to get a preview of one of the conclusions, but when we look at the nitrogen measurements, the pattern looks pretty much the same. Most of the variance is among sites, but then there's this component among treatments that we can look at to see how what the practices are doing. So what is driving these differences among sites? When we look at soil organic carbon, we can see there's a significant negative relationship between carbon and annual mean temperature at the site. So sites with a colder climate have higher soil carbon on average than sites with a warmer climate. Similarly, with clay content, we can see there's a significant relationship where soils on average with lower clay content have lower soil organic carbon. Soils with higher clay content have higher organic carbon. And this isn't really a surprise for soil organic carbon. There's plenty of research that's been done before on looking at these, regional or continental or global scale patterns of soil or organic carbon. When we extend that to the rest of the measurements, we do see pretty much the same pattern. So these site factors do affect the absolute values of these carbon measurements. So we looked at 5 pretty simple ones that you might use to characterize a site. So the sand content, clay content, pH, mean annual temperature, and precipitation. And we've also added irrigation to that for those sites that have been irrigated. And we can use a multiple regression model to try to take into account how much all of these factors can explain simultaneously. And so just like soil organic carbon, all of these indicators show that negative relationship with temperature. So higher values for all of these carbon measurements at the colder sites. And we generally see an effect of precipitation and clay as well, with higher values of these carbon measurements when clay is higher, where precipitation is higher. And using just these 5 simple to measure site characteristics, we can predict anywhere from around a quarter to about half of the variation across sites. Once again, the nitrogen measurements have a similar story. So this negative relationship with temperature, generally a positive association with clay and a positive association with precipitation. And then somewhere, in this case, just under a quarter to just over half of the variance in the data set can be explained with these 5 variables. So we know that site needs to be considered when interpreting any of these measurements. The absolute values vary from site to site, but some pretty basic information about soil texture and climate should be sufficient to kind of put these sites in context. So that's the across-site variability. What about the within-site variability? And so within-site variability in terms of NAPSHM is mostly related to the management, so the treatments at these sites. And we looked at these treatment effects by narrowing down the data set and focusing on sites that had paired treatments that were identical, except for one of the 6 soil health practices shown here. And so this graph shows the percent change in soil organic carbon associated with adopting one of these practices. So the circle in the middle represents the predicted change in soil organic carbon, and then these whiskers here represent a 95% confidence limit. So when those whiskers don't cross the 0 line, so for tillage, cover crops, organic nutrients, and residue retention, we'd say there's a significant increase in soil organic carbon with these practices, whereas these 2 associated with crop diversity, we don't see a statistically significant effect. And this is something that we see consistently throughout the measurements. So these rotation diversity, which compared treatments with only grain crops to those with some other type of crop, typically a legume or a brassica or a cotton, we don't typically see an effect on any of the measurements. And then crop count, which compared monocultures to a rotation that had 2 or more crops in it. And then we typically do see a response to these other 4 indicators. So for when the treatments represent a decrease in tillage, and this could be from a reduced tillage to no-till or from a disc to no-till, any type of reduced tillage, on average, we saw about a 10% increase in soil organic carbon. And we see similar magnitude changes from these other practices. So by including cover crops, by substituting organic sources of nutrients, so manure, compost, biosolids, for at least some commercial fertilizer, or retaining more residue compared to removing that residue, we see an increase of somewhere around 10 to 25% in soil organic carbon. When we expand that to the other indicators, we see a similar pattern. So these crop diversity indicators, we never see a significant positive effect for any of these 6 measurements related to carbon. For these other 4 practices, residue retention, organic nutrients, cover crops, decreased tillage, we generally see a positive effect like we did for soil organic carbon. So for the POC, for water extractable organic carbon, for the 24-hour potentially mineralizable carbon, we all see that increase. The 96-hour potentially mineralizable carbon, we actually see it's a little less sensitive to management than the other indicators. And then the enzyme activity is actually relatively sensitive as well. What about the nitrogen indicators? Well, just like as we saw earlier, we see a similar pattern that we saw for carbon. So never a positive response to these diversity indicators. And in some cases, we actually see a significant negative response. But then for residue retention, organic nutrients, cover crops, decreased tillage, we generally see a positive response for most of these indicators. Once again, there's some indicators where for some practices, we don't see a statistically significant response, but generally the same pattern as carbon. We saw earlier that site can have a big effect on the absolute values of the measurements, but another important question is whether site affects the response to management. So are these measurements applicable just in certain soil types, certain regions with certain climates, or are these measurements giving us the same answer kind of across North America? And the way that we looked at that was using these response ratios again, so looking at the response to a practice. And we only did this with tillage because we had most sites to look at. The other practices, we had a lot fewer sites. But so the y-axis here represents that response to decreased tillage. So above 0 means a positive response to decreased tillage, and below 0 means a negative response to decreased tillage. And each one of these points represents an individual site in NAPCHM. And then the x-axis here is the amount of water these sites get from precipitation and irrigation. And so for soil organic carbon and for water extractable organic carbon, there is no effect of these water inputs. So on average, as I mentioned before, there is a positive response to decreased tillage, but we can't say that at wetter or drier sites that the magnitude of that effect was different. For the other 4 indicators, for POC, for the 2 potentially mineralizable carbon measurements and the beta-glucosidase enzyme, we do see that there's generally an increase in the response to decreased tillage with precipitation. But what I'll point out is if you look at where these best fit lines cross 0, we would only predict a negative response of these indicators at the very driest sites. And so we feel generally confident that these indicators are widely applicable across the sites. And I'm showing you the plots of precipitation and irrigation because those other 4 site characteristics, so sand and clay content, pH, and temperature, didn't see any relationship at all with these responses to decreased tillage. So generally, these responses are appearing kind of across North America. So we had all of this data, but we needed to decide how to choose the recommended measurements. For carbon, I mentioned already why we rolled out the loss on ignition and microbial biomass. But another factor that comes in if we want to use these measurements at scale is the cost and availability. So we contacted 7 commercial labs that we knew around the country just to get some rough estimates of whether they did these measurements and the price. And you can see that it ranged from only 2 that did the enzyme activity up to 7 for the permanganate oxidizable carbon and the soil organic carbon. And you can see there's also a wide range in price from as cheap as about $14 up to about $45, but it varies a lot from the labs. But so in general, we would prefer a cheaper and more widely available measurement. Another way that we can start to rule out measurements is if they're redundant with each other. And it turns out all of the nitrogen measurements were really strongly related to similar carbon measurements. And so whether it's looking at total soil nitrogen to carbon, the water extractable nitrogen to water extractable carbon, the 2 enzymes, the potentially mineralizable nitrogen versus potentially mineralizable carbon, all of them were so highly related to each other that we don't get that much additional information by measuring both nitrogen and carbon indicators. So how do we put all of this together? So all of these measurements have potential to detect, to be used for soil health evaluation, but we can't recommend all of them. We want to take into account, are they relatively direct measurements of these pools and processes of the carbon cycle? Want to have a measurement that's easy for people to collect and easy for people to understand without any methodological complications. We can eliminate measurements that are redundant with each other. We can eliminate measurements that aren't as responsive to the soil health management. And so when we take all of these, and we can take into account the cost and availability of them. And so when we take all of those factors into account, for carbon, the indicator that best met all of those selection criteria was soil organic carbon. Can be directly related to that pool of carbon in the soil, and it's easy to interpret and understand. But we also chose the 24-hour potentially mineralizable carbon indication because it's a pool of carbon that can be readily used by microbes. And so just to summarize, when we're choosing these carbon and nitrogen measurements, the location matters for the absolute values. All of these measurements generally increased in response to soil health management, except for the crop diversity managements. The nitrogen and carbon dynamics are so tightly linked that we feel we don't need to measure those nitrogen indicators. We have many good measurements, but some are cheaper and easier to interpret or more widely available. And so for a minimum suite of indicators, we recommend the soil organic carbon and 24-hour potentially mineralizable carbon. So with that, I'll thank you with all the people who helped with this and our funding sources.